It's really a pleasure to be here and to see all these great people, immunologists I admire. And I am myself a philosopher of science, and I'm going to explain a little bit the way we try to work in the immunology lab in Bordeaux, putting together immunology and philosophy. So my talk is about extended immunity, which I'm going to define, and its consequences for cancer. And I know it sounds like a huge title and a huge topic, and indeed it is. And in a way, I will make things even worse by having an even bigger claim or broader claim, which is that immunologists should pay attention, and they do to some extent, but sometimes this is something that should be strengthened. I think they should pay attention to the diversity of the activities of the immune system and try constantly to think about those activities in an evolutionary context. And I'm going to explain what it means. But first, I want to start with this question of cancer immunology. I think that the main underlying question of cancer immunology is, how does the immune system interact with cancer? And this is a proximate question. And there's also an ultimate question, which is an evolutionary question, which is about why exactly immune systems interact the way they do with cancer. And I think that that sounds probably obvious, but I think that characterizing the interactions between immunity and cancer and exploiting their potential in future research requires a precise and up-to-date understanding of the immune system. And I think that oncoimmunology rests on a misunderstanding of the immune system, or rather on a very partial view of what the immune system is and what the immune system does. So basically, my very general suggestion is that current oncoimmunology and current immunotherapies in cancer are just right here in the iceberg. And there is this incredible big continent that has to be explored if we want to understand better oncoimmunology and immunotherapies in cancer. So that's a very general claim. I know it's pretty vague, but I'm going to try to make that a little bit more precise. But basically, that's the idea. We're going to use extended immunity, which I'm going to define, as a way of thinking more broadly about cancer immunology, and how it works and how it could work in therapeutic practice. So that's the idea of an extended immunity. And my conceptualization of the immune system is that the immune system is a controller of the organism's cohesiveness. Admittedly, an incredibly vague definition of the immune system. I don't think there is any precise definition of the immune system. So I think we're all in the same boat here. If you want to define the immune system, either you will say something specific and wrong, or you will say something extremely general that might be true, but extremely general and vague. And I think this is really the sort of challenge that we all have to face. More about this later. So my main thesis in this talk is that oncoimmunology must be reshaped based on an extended and more adequate view of what an immune system is, namely a controller of cohesiveness. For this talk, I'm going to present very quickly our philosophical, conceptual approach to biology and medicine, because I think it's important to contextualize a little bit what we're trying to do, which is very similar to what Martin presented this morning, the idea of putting together philosophy and immunology, which is, of course, something which is represented by many people in this room. Basically, you have several philosophers of immunology in this room, which is something pretty rare because there are not that many of them. And one reason for that is that, as you know, especially if you are an immunologist, people are interested in immunology, attracted by immunology very often because they are said that it's extremely complex. It's very complex. So when you're a philosopher and you want to study biology, the first thing that biologists and medical doctors will tell you is don't go to immunology. It's the worst. It's super complicated. And I think that all of us here in this room thought, oh, that's interesting because it is so complex and so intricate. Anyway, I'm going to explain how we do that. Then the traditional view of the immune system, the view of the immune system in current oncoimmunology, my own view of the immune system and how this view could help reshape oncoimmunology. To be clear, the fifth part will be by far the longest part of my talk because this is a way I want to get. So all the rest is going to be pretty quick. In terms of methodology. So a few years ago, 3 years ago, I coordinated this paper Martin alluded to, Why Science Needs Philosophy in PNAS. And we gathered different people, philosophers, historians of science. He's a Chan, for example, is a major historian of science at Cambridge. And people you probably know, like Alberto Mantovani, Margaret McFarlane, and a few others. Carlo Rovelli, as you probably know, is a major theoretical physicist. And we decided to explain why we thought that there was this misunderstanding about the connection between philosophy and science. And this is pretty much exactly what Martin said. Basically, the very general idea is that it's only starting from the 19th century that there was this divorce between philosophy and science. So pretty much all the history of philosophy is about talking to science or doing science and philosophy at the same time. Suddenly, because of the speciality, not suddenly, but quite suddenly, historically, you have suddenly a divorce between this philosophy and science because of the specialization of knowledge. So people realize they cannot be any more universal scholars. But this is fine. I mean, it is true that we cannot be universal scholars anymore, but collectively, we can continue this work of putting together philosophy and science. And that's basically the idea, just continuing the long tradition of philosophy being with science being a part of philosophy being the same thing as science, etc., etc. We can talk about the details, but that's basically the idea. So what we show in that paper is that there's an urgent need for stronger collaborations between scientists and philosophers. We also give 4 very specific examples of recent contribution of philosophers to science. These contributions are not necessarily huge contributions. You know, we're not pretending that the whole field is going to be changed by what these philosophers have done. But we also have to acknowledge that most scientific contributions are modest scientific contributions. You know, this is very weird to ask the philosophers to be major changers in the field when they're interested in the field. Science is about small changes all the time. Science is very incremental most of the time. And what we show in this paper is that sometimes philosophers have made suggestions, for example, in cognitive science, in neuroscience, they have made suggestions that have become major part of current science. And also, we show that the kind of interventions that philosophers do have mainly to do with conceptual clarification, critical assessment of scientific assumptions or method, formulation of new concepts and theories, and connections between different sciences. This is one thing that philosophers do very well. They know very little about different fields. So connecting the fields can be easier when we know a little bit about different fields. And this is something that we can do because we are less specialized most of the time than scientists, even though philosophers of science become increasingly specialized with time. So the methodology we try to implement in the lab is really this philosophy in science idea that I try to characterize in recent years. This is an old picture, unfortunately, you can say that because I look much younger than I am. And this is a picture where we had the different teams of our lab. So our lab has about 100 members. So intermediate size lab, but still a big lab. And we do some very traditional immunology, for example, about autoimmune diseases, about cancer immunology, about inflammation and cancer. Also, original work about gamma delta T cells. Judy mentioned gamma delta T cells before, and we are one of the labs that has some strong specialization on gamma delta T cells. And we have this very different team, which is the conceptual biology and medicine team. That's now I'm the co-PI with my friend Maël Lemoine. So the idea is to have embedded philosophers who are in the lab and work constantly in the lab. And we think that that makes a difference to the kind of philosophy we do, and that makes a difference to the kind of science that people do in the lab. That's basically the idea. And we think that being interventionist is extremely important. So about 70% of the publication of our team are in science journals, not in philosophy journals. And just imagine that it's extremely challenging when you're a philosopher to submit your paper to a science journals. They have no idea you're a philosopher. And you try to convince the editors and then the reviewers that your paper is worth publishing in a science journal. So we like this challenge. We think it's very important because our perception is that we should try to make a difference to science. As I said before, it can be a small difference, but if you want to make a difference to science, you have to talk to scientists. You have to convince scientists that this is interesting, and therefore you have to publish in their own journals. So that's currently the team. We have 3 MDs. We will have in a few weeks 3 MDs, 3 biologists, 6 philosophers, and 1 administrative assistant. And so you see, interdisciplinarity is really there in the team, in addition to the team being part of a big immunology lab. What do we work on? We work on conceptual immunology, which is a vague and general idea, but we explore specific concepts in immunology. We work on cancer, especially on cancer and evolution. We work on aging. We work on computational methods in biological medicine, and also we work on the microbiome. Just to show you that we are truly embedded in the lab, here is your favorite philosopher, Martin Zach, in our lab a few years ago. And you see, this is really like, here it's the animal facility, not in the lab, and here in our lab in Bordeaux. Here are some examples of recent work we've done. One was about the redefinition of immunological memory as a multi-realizable and multidimensional process. This is something I did with Louis Dupasquier, who is a specialist of evolution of immune systems. This is something we did more recently about the concept of exhaustion. And this is, again, something where we believe that defining concepts as precisely as possible can be useful. There is this idea that philosophy would be about vagueness and science would be about precision. In fact, a lot of philosophy is about determining to what extent we need to be precise when we use concepts. Sometimes we have to be very precise. Sometimes a little bit of looseness can be useful. And one of the things that we philosophers of science try to do is to determine in a given science, when should we try to be more precise when we use a term? And exhaustion, I think, was a good example of that. Exhausted T cells are everywhere. What you mean by exhausted T cells is a different matter. And when people have started to say, oh yeah, what happens in cancer is that you find exhaustion exactly the same way that you find exhaustion with viruses, for example, we are like, yeah, but let's try to see more precisely what you mean by it. You mean exactly the same thing by exhaustion in that context, cancer, and in that context, viruses. And it turned out that there are many differences. And we try to explain this thinker about three kind of definitions of exhaustion, including functional ones, which we believe are not equivalent. And this is very important to clarify. Anyway, we also did some work on cancer-associated microbiota in terms of diagnosis, causality, and therapeutic. This is something we did recently with Rob Knight at UCSD. And we've worked quite intensively about the microbiota and immunity in my ERC project that ended 2 years ago. Philosophy in science, the way I defined it before, is the idea that philosophers should not just intervene on the interpretation of scientific results, but they should intervene upstream. They should intervene in the moment when scientists gather in the lab and say, what should we do? What should be next? And we try to make suggestions in terms of experimental programs. To be honest with you, very often it doesn't work in the sense that our suggestions are crazy. I will give you some examples in a few minutes of the kind of stupid ideas we can have. But the idea is that a little bit of originality, a little bit of new ideas, a little bit of thinking differently sometimes does lead to novel experiments. And this is something we've done recently in many different contexts. Again, I'm certainly not pretending that we have made sort of game-changing contributions. I'm just saying that doing that kind of intervention with the scientists and trying to redefine the experimental programs of scientists in a lab is something that we really wanted to do, and we believe that it can be extremely interesting. With that, I can turn to this idea that the immune system is generally defined in a certain way, and as we will see, this impacts oncoimmunology. So the way in which we think about the interactions between cancer and the immune system. In the traditional view, the main activity of the immune system is defense via elimination. So this is what can be called eliminative defense. This is the idea that the immune system is capable of detecting something and eliminating that something when needed. The detection is mainly based in the traditional view on this idea of self and non-self that was beautifully suggested by Burnett, starting from the 1940s. And this is something Bart has been in this room, has been very interested in, how this idea of self, non-self was built and how it was changed over time by different researchers. And is it still alive today? And this is very interesting because my impression is that most immunologists are now very careful with self, non-self. They would not see themselves as extended the views of Burnett, I think. I think they consider that a lot has been changed to this view. Still, my own impression, and it's not just an impression, this is something I try to demonstrate, is that we're still living in that framework of self, non-self. This is still there. So even though you can find all sorts of explanations about infectious non-self, for example, or danger model, or the self non-self is still very much there. A great example for me was when CRISPR-Cas started to be described as an adaptive immune systems in prokaryotes. Self and non-self was everywhere. So I I reached these people and said, why are you saying this is a self, non-self detect? Because it detects pathogens. You know, it detects phages. Therefore, it detects pathogens. Therefore, it is a self, non-self detection system. And if you're like, is there any way in which that system could detect something that would be self, you know, that could react to the self? And they were like, initially, they were like, no, it doesn't make any sense. It's a system of detection of pathogens. And then, of course, it does. Of course, it does sort of autoreactively respond to some components, because all immune systems do that. What I just want to say is that the self, non-self is still very much there. I think almost everywhere you look at in immunology, you will find the self, non-self still there. The main components of the immune system, according to the traditional view, is adaptive immunity components, mainly lymphocytes. And in terms of phylogeny, Burnett was very clear, for example, in his Nobel Prize lecture in 1960, that immunity is about vertebrates. Immunity is about higher vertebrates. This is very clear for Burnett. And of course, I think this is wrong, and I think this is part of this traditional view. There were many critiques of the self, non-self. I think that, to me, it's a little bit of a shock when I teach to young immunologists to realize that Polly Matzinger almost disappeared from what people know. And I think that is a problem. I don't think Matzinger was right, but I think for many people in this room, Matzinger was very useful. The way in which he suggested the danger knowledge, first as a critique of the self, non-self, and then something that was refined progressively as something which is not so much about danger, but about damages. I think she was vague about that. But if you take the danger model to give a theory about damages, I think this is a pretty interesting theory that is not unrelated to what Judy and other people do in their work. I think there is really something about that that tends to be a little bit underestimated at the moment. The self, non-self has had many competitors, and this is interesting to, I think, to study those competitors. Anyway, so that's the traditional view. That traditional view impacts current oncoimmunology, I think. I can be quick on this because you probably know that already, all of you in this room. The idea that the immune system can control cancer is very old. It's been often associated with William Coley, but in fact, it's a very frequent idea at the end of the 19th century. So Paul Ehrlich, for example, does talk about the connection between cancer and immunity in interesting ways. So in the case of Coley, it was the idea of using bacteria to fight cancer. He's sometimes called the father of immunotherapies. And in that case, this is, as you understood, pathogen-mediated immunogenicity. Immunological surveillance is a concept that was put forward by Lewis Thomas and Burnett. It is often said that Burnett invented the idea. No, Burnett was super clever. He was going to all sorts of conferences and he was very quick to publish. So Thomas is the one who suggested the idea and Burnett picked it up and published more quickly than Thomas. So for all of us who pay attention to the history of science, it's important to emphasize that Lewis Thomas was the one suggesting the idea first. But anyway, it doesn't matter. The very general idea is that immune systems have evolved to detect cancer. This is a super strong idea suggested by Burnett, and in a sense, creating some sort of conflicts with the self, non-self view, as I'm going to explain in a second. So that's really the idea that immune systems can detect cancer, and there was this strong selective pressure on immune systems to detect cancer. Remember, for Burnett, we're talking about adaptive immune systems. There's nothing to say here about the innate immune system being able to detect cancer. We'll talk about that later. Okay, so that's immunological surveillance. But why exactly would the immune system recognize and eliminate cancerous tumors? After all, tumors are mostly endogenous. They are mostly self. So should they be seen as self or non-self? And as you can imagine, this was a major question for Burnett and people working around Burnett and trying to answer Burnett's views. Immunological surveillance and the self, non-self view say, no, of course not. Tumors are not self. They are altered self, modified self, and hence all the vocabulary of neoantigens and neoepitopes, which is still very, very present in our science today. And that is very interesting from a philosophical point of view, because as I tried to show years ago, this is purely circular. This is purely topological. So you see that there are some immune responses to cancer. And therefore, you try to define as much as you can the target of that immune response as a form of non-self. So the very idea there was, what is non-self? It is what triggers an immune response. And what does trigger an immune response? It is non-self. So what would be very interesting in that case would be to say, why exactly those alterations count as non-self? And why do not other alterations, think about puberty, for example, why do these alterations not count as non-self? Do they in some circumstances? Then explain to us, what are those circumstances? So this is a very interesting example where you have something that is just made as a new vocabulary to save the theory. You know, this idea of altered self. I'm not saying it's wrong. I'm saying it's clearly conceptually flawed. And you have to be much more precise if you want to say why there is a response to cancer. Of course, immunological surveillance was a certain period of time. I've shown the book of Burnett in 1970. Okay. So that idea was then abandoned, as you know, for several decades, and then it came back under a different form because the experimental models that were used by Burnett were criticized, and then other models emerged, and then many people, including Schreiber, for example, and the work around Robert Schreiber, was very important to demonstrate this idea of three phases of immunoediting. Elimination is pretty much the same as immunological surveillance as Burnett started or conceived it. Then you have a phase of equilibrium where some cells which are resistant to these immune cells are going to survive, and then they can escape the action of the immune system. So we have here a very interesting explanation of why the immune system can both contribute to the destruction of the tumor and also contribute, for example, to very complex and damaging metastasis, for example, in that kind of cases. So what is the immune system in current oncoimmunology? So we see here the traditional view. So to what extent does the traditional view impact oncoimmunology? I think that in current oncoimmunology, the idea that the immune system's main activity is to eliminate this sort of eliminative defense, as I said before, I think this remains very strong. So for example, the idea that the immune system is different to the organs of the elimination of the targets recognized by the immune system. Applied to cancer, we would have the idea that the immune system can recognize and eliminate cancerous tumors. And this is something that is a little bit everywhere in the literature, and that immunotherapies are aimed at harnessing or boosting immune responses. This is just one example of that, but this is everywhere in the literature. CAR T cells, for example, are very often conceived as, a perfect example where you take, in some cases, take patients' T cells, they are engineered in the lab so that they can recognize cancer-specific antigens and then re-inject them into the patient. And I'm certainly not saying that this is not a promising strategy. I think this is a very promising strategy. What I am saying is that those kinds of research are based on the idea that we should boost the immune system so that it can destroy the tumor. This is science in 2013, cancer immunotherapy, T cells on the attack. If you look at this NIH movie, immunotherapy, how the immune system fights cancer. And I think there's some ambiguity here, something which is not necessarily the way in which we should think about the interactions between the immune system and cancer. But my point here is very simple. It's just that the main activity of the immune system as conceived of by current oncoimmunology is that the immune system can and should eliminate tumors. And we can help the immune system to do that. The detection is mainly based on the idea of this altered self concept that I presented before. What are the important components? It's mostly about adaptive immunity when you read oncoimmunology, but there is this very big change. So some people have been interested in that for decades. They have been interested in the way the innate immune system plays an important role in cancer. Many examples of people having done that. One is Alberto Mantovani. I mentioned him before. So this idea that, very strong idea in the initial work of Mantovani, this is clearly the idea that the immune system, most of the time, helps the tumor. And then, of course, so if you read the Cancer Inflammation of the Lancet in 2001, the idea of Mantovani, which then was not exactly confirmed by, the Nobel Prize about immunotherapies, and is that the immune system is mainly promoting tumors instead of destroying tumors. And from the point of view of M2 macrophages, for example, which is something we will hear about later today or tomorrow, this really makes sense. So the idea that in my sort of iceberg that I presented before, one thing that I think everyone agrees on in the domain at the moment is that a lot about immunotherapies is going to come from innate immunity. This, I think, is a pretty consensual point. Still, a lot of oncoimmunology and immunotherapy therapies is based on adaptive immunity. What about phylogeny? People doing oncoimmunology, most of the time, they consider that immune systems exist only in vertebrates, or most of the time, in fact, they just don't care. So there will be no question about, should we investigate how immune responses to cancer occur in different organisms, for example, in vertebrates? This is a question which is very rarely raised, but of course, I will present some exceptions. So my own view of the immune system is based on this very general idea that we should extend immunity because we should realize that immunity is much more than defense. And we should realize the different ways in which immune responses are produced in actual practice in organisms. I recognize this is a super vague idea, as I said before, but I've suggested, for example, that the immune system should be conceived of as a controller of organisms' cohesiveness, especially at the level of tissue organization. I insist again, this is extremely general. What I want to do with that is to suggest a different view of the immune system that could be helpful in different contexts, including the context of cancer immunology. So that's the aim of what I'm saying here. In my view, the main activity of the immune system is to control cohesiveness in the organism. It means that immunity is as much about repair, development, metabolism, for example, as it is about defense. I'm not underestimating the importance of defense. I'm not saying that defense was not important evolutionarily. What I'm saying is that if you focus on defense only, you're going to miss a lot of what's going on in current immune systems. If you don't pay attention, for example, to the incredible evolutionary pressure, selective pressure that it is to be able to develop properly and to repair yourself constantly, you're going to miss many of the functions and dysfunctions of current immune systems. So a view that is based only on defense is going to be biased because it is too narrow. That's the main message. I think that in terms of detection, immune systems detect sudden changes in targets. I'm going to explain that in a second. I think that in terms of components, innate and adaptive immunity are both very important, but I give priority, and I can explain later what I mean by priority, to the innate immune system. And my view, which is, I think, now much more widespread than a few years ago, is that there is no organism without an immunity. This is something which is often to critique. I mean, this is something that is really at odds with what people have said for decades. But now I think it's extremely difficult to defend the idea that prokaryotes do not have an immune system, that plants do not have an immune system, that invertebrates do not have an immune system. And this goes back to Mihai's point about the kind of selective pressure that it is to be able to respond to different challenges, including pathogens, but not only. Then the question of memory becomes an additional question here. But the very question of, can you have a sort of immune system, I think should be replied very positively for all organisms. Clearly for all multicellular organisms. I had many discussions with microbiologists. They claim that sometimes bacteria don't have anything immune-like. I do think it depends on how you define immune in those cases, but at the very least, I would claim that all multicellular organisms have an immune system, and I would be very much tempted to say that it's much more general than that. So how this view could help reshape oncoimmunology? This is what the final part of my talk will be about. I think there are three main dimensions of this reshaping of oncoimmunology. If you think about the immune system as a controller of cohesiveness, I think it impacts at least three questions. One is about detection. How does the immune system detect a challenge? One is about activity. And here I'm going to talk quite a lot about repair, tissue repair more specifically. And also, it has some consequences in terms of phylogeny. So I'm going to focus more on this part, but I start with this, detection. So my very general view about this question is that immune systems detect disruptions of cohesiveness, especially at the tissue level. I think that immune systems have been selected for evolution to detect sudden disruptions of cohesiveness. The very general idea being that the control of cohesiveness consists in a control of the speed of change in targets surveyed by the immune system. The idea being that immune components constantly interact with what's going on in an organism, and they will detect strong changes, quick changes. In terms of the discontinuity theory of immunity that I suggested several years ago with some colleagues, including Eric Villiers, who is a specialist of NK cells, what we suggested is that an effector immune response is triggered by the sudden modification of the molecular patterns with which immune receptors interact in a given environment. Applied to the example of antigens, that is the main example that we used in that paper, we suggest that what counts for an immune system is antigenic variation with regard to time. I am certainly not saying that this theory is true. I'm certainly not saying that this theory answers all the kind of questions I raised before. What I am suggesting is that one way we can conceive of the action of the immune system is that it doesn't care at all about something being self or non-self. It cares about detecting strong and rapid changes in the organism. A cancer, as I'm going to say in a minute, can be part of that. And of course, again, those things are going to be species dependent. Those things are going to be very much context dependent, as explained here. So, of course, a discontinuity in your gut is not the same thing as a discontinuity in your brain. But the very general idea is that the immune system is constantly monitoring what's going on in a given organ, in a given environment, especially in the tissue, and it will react to strong disruptions of this something going on there. Okay, it's vague, but I don't think other hypotheses are less vague than that. And I think this is interesting to see things in this way. So in that paper in Nature Reviews Immunology, we give different examples of that. I don't want to go into the details. If you're interested, we can talk about that. What is the consequence of this view for cancer? Well, the idea is that theoretically, the rate of alteration for the discontinuity theory of immunity is much more important than the concept of altered self and the concept of neoantigens. If I'm right, then the source of the antigen is that self or non-self doesn't matter as much as the speed of change. So the prediction of the discontinuity theory of immunity is that slowly growing tumors and persistent tumors will induce tolerogenic responses, while rapidly growing tumors will induce effector immune responses. And this is, of course, something that can be tested in the lab, though it's not as easy as it sounds to test that kind of idea. If we manage to accelerate the rate of antigenic change in a tumor, we will induce a stronger effector immune response. That's part of the suggestion. So again, I'm not asking you to buy this. Maybe you have strong objections. Myself, I have many objections to that view, which is my view. Over the years, I've started to raise all sorts of questions with regard to this view. I just wanted to say this is one way of thinking about what it would mean for an immune system to be a controller of cohesiveness at the tissue level. You're not going to detect all sorts of changes. If you detect every changes, there's no way in which the organism itself can develop, evolve, change, et cetera, et cetera. So the very general idea is that this is one way of seeing how changes could occur in the organism, and sometimes lead to an immune response, an effector immune response, and sometimes not. Okay, now I focus on this question of activity. I think that it is now relatively clear that immune responses go well beyond defense. So in my book that I mentioned previously, I gave some, I think, important example of that. Immunity is about defense, but it's also about clearance of debris, for example. It's very much about repair, about development, about the maintenance of tissue homeostasis. This is a point that was explored by Rankin Artist in Cell in 2018, beyond host defense, emerging functions of the immune system in regulating complex tissue physiology. And much more modestly, we suggested in this paper that immune components are absolutely crucial for repair at all levels. So you can imagine how ashamed I am to present that in front of Judy. But I mean, we think that that aspect of immunity is both well-known and underappreciated. If immunity is about repair, then what? And we believe that the consequences are pretty strong. The analogy I like is with Pradeu, the game. Of course, a wrench can be used to kill. Of course, immune systems kill. Of course, immune systems destroy. But why would we think that this is the function of the immune system and that this is what immune systems have been selected for through evolution? I think this is a major mistake. It doesn't mean because a wrench can kill that the main function of the wrench is to kill. Again, not underestimating at all the importance of defense and elimination of pathogens. But all that is a question of different activities, different functions going together. And if you pay attention to that, then you are led to this idea that we should constantly rethink immune responses from an evolutionary perspective. And I admit that some people do that, but most immunologists I know are not interested in evolution at all. And they don't think about those questions, I think, intensively enough. It's more like immune systems have evolved for this, for that. Yeah, but try to get a comparative perspective and try to understand the diversity of selective pressures that were exerted on immune systems per time. I think that is a major and promising avenue for research in immunology. It is always useful to think what immune systems do in present context, in the context of what they've been selected for through evolution. What is the evolutionary history of immune systems? We know very little about that. Which selective pressures with which respective weights? I think pathogens, development, repair have been major selective pressures. And just to give you one example, which I think is extremely important, I think cancer was a very minor selective pressure. We can talk about that. Some people think it's a major selective pressure. I think this has been minor selective pressure, which means that the immune responses to cancer that we see today are a byproduct of something else. And if they are, it changes entirely the way you're going to interpret the data and also the way in which you can interpret adverse effects, for example. So if you believe the immune system should destroy cancer because this is what it's been selected for, you're going to be perplexed by some of the reactions that you see in immune systems today when they respond to cancer. If you pay attention to the fact that immune systems have been selected for many different activities, including repair, then a lot starts to make sense. So my main focus in terms of immune activity here is going to be repair. I told you about this, the very traditional view of the immune system. The immune system is about eliminative defense, and this has consequences for cancer. My crazy idea of the day, I have many crazy ideas, and this is clearly one. And I apologize, this is very speculative, and the idea is just to submit a very speculative idea to this audience instead of saying, oh, look, this is truer, or this is more adequate than other views. My suggestion is the following. What if the immune system could be manipulated not to attack, but instead to repair the cancerous tumor? And I'm going to explain that right now. There are important similarities between cancer and tissue repair. This is something that has been noted by Reshaping and several others in the 19th century. And then there is this paper by Dvorak that was published in 1986, which is a very famous paper. I don't think people read it that much because the content is very different from what people say when they cite the paper. But the paper is very interesting and suggests that tumors are wounds that do not heal. And then Dvorak wrote a sort of update in 2015 and another update in 2019, which is very useful. So there is this idea that there are many similarities between cancer and tissue repair. This is an idea that has been explored by people like Schaeffer and Werner. Cancer as an overhealing wound, an old hypothesis revisited. and also something that has been insisted upon by Mina Bissell, among many others in recent work. So the idea is that cancer appears as a never-ending, never-resolving healing process. And now we put together, so we said that cancer and immunity have a lot in common in terms of, they have strong interactions. Then we say that immunity is central to repair. And then now the idea is to put together cancer, repair, and immunity. The idea is that wounds and inflammation can create tumors, and tumors can create wounds and inflammation. I suggest that in many such cases, the immune system will do what it primarily does, which is to repair. Therefore, immune-mediated repair can be perfectly normal and still contribute to cancer because the tumor is seen as a wound and or co-occurs with a wound. So the very general idea is that in those contexts, if the wound is there, or if the cancer is perceived as a wound by the immune system is going to repair. Immune-mediated repair can also be dysfunctional and or co-opted by the tumor, but I don't think it is necessary. When people start thinking about those issues, immediately they will say, oh yeah, this is because the tumor manipulates the immune system. I think this is one option, but we should be careful with seeing everything through the lens of manipulation by cancer. I think very often the immune system is not really manipulated. It's just doing what it does. It repairs because it looks like a wound, or it is coming with a wound. In some cases, asking why doesn't the immune system kill the tumor and how can we promote the immune killing activity is doomed to fail. We presuppose a function, elimination of a foreign entity, and the associated dysfunction, and I think this is wrong. It's not foreign, and the immune system is not eliminating, it is repairing. So I think there is a key avenue here, which is to understand and intervene on immune-mediated repair by promoting or inhibiting that response, especially through neutrophils, which are here, but I don't want to go into the details. So can we immunologically repair cancerous tumors? I have one hypothesis, which is that some cancers result from abnormal tissue repair, either dysfunctional tissue repair or perfectly functional repair, but in an abnormal context, which is not the same thing. So in one case, you have problems with your repair mechanisms. In another case, repair mechanisms are perfectly fine. It's just that the context is pathological. And one question for you is, how could we demonstrate this hypothesis? The second hypothesis is that the immune system has a major influence on how abnormal repair generates cancer. And again, I'm asking, how could we demonstrate that? Of course, maybe you think it's crazy, and if it's crazy, maybe we don't need to spend too much time demonstrating the hypothesis, but if you believe it could be interesting, I'm interested in determining how we could demonstrate that. And the third is probably the most important. One can manipulate the immune system to make it repair cancerous tumors. So how to demonstrate that? How in practice the immune system could be manipulated for this purpose? My suggestion includes something you necessarily have in mind at the moment, which is blocking type 2 response. And this is already done, and this is perfectly fine. I think this is important. In some cases, what we will want to do is to block the type 2 immune response, at least in some contexts. But what I'm suggesting here is a little bit broader than that. I have the impression, and maybe this is where this is completely crazy and completely wrong. In some cases, I think we should promote tumor repair by stimulating the type 2 response instead of blocking the response. So the very general aim would be to try to achieve the resolution phase, because the problem with cancer would be that we do not reach the resolution phase. But of course, there are several central questions when you suggest something like that. When should we do that kind of interventions? Would it result in excessive fibrosis, which is a huge risk with cancer? As you know, very often tumors are fibrotic, and then many problems occur, including the impossibility for immune cells to get close to the tumor, which of course would be a disaster. So I'm certainly not suggesting that we should test that kind of things in humans. I'm saying maybe we could think about that and find suitable models to try those ideas. And I have this collaboration with my friend Marie-Elise Truchte, who is a medical doctor specialized in fibrotic diseases at the hospital in Bordeaux, and we are trying to explore that idea. So I see two conceivable moments for interventions in terms of immunologically mediated repair in cancer. One would be very early in tumorigenesis, and that would be the idea of preventing the vicious circle of cancer-related or cancer-inducing never-ending damages. I don't know if it's doable, but that's one possibility, conceptually at least. And another intervention would be in late tumorigenesis. And that would be the idea of normalizing the tissue for tissue remodeling before dissemination happens. So in one case, you try to intervene very early. In another case, you try to intervene very late. So we have two papers in preparation on this issue. One is historical, the other is conceptual, and I can see that I don't have much time. So I just wanted to say that Judy has this great paper, among many others, which is about parasites and tissue repair. I think current research on tissue repair is absolutely central. It's changing the very way in which we conceive the immune system. And when I read sentences like that, I'm immediately very interested. Over evolutionary time, it seems that mammals have learned to recognize the presence of potentially damaging large multicellular organisms and initiate a response that largely resembles a reaction to tissue injury. And I'm suggesting that maybe it's even broader than that. And then we have to evaluate, is it good to have a broader conception, or is it better to have a more precise conception? As I said before, my impression is that it would be beneficial to think about the immune system as a system that can detect damages or disruptions of cohesiveness that could include cancer. And that would mean that tissue damage is just a part of this way in which you can disturb or disrupt the organization of a tissue. I don't have time for this. The very general and simple message is that I've been very interested in cancer repair and immunity in Drosophila. I think it's a wonderful model. I've interacted with different people recently, including David Builder, who is at Berkeley. I think this is a wonderful model. One reason for that is that, for example, you can have 1,000 Drosophila when you do your experiment. So I've been interacting for a long time with Bruno Lemaitre in Lausanne, but now it's different. It's a context in which the idea would be to test the connection between cancer repair and immunity in that model. And I think this could be a great model to test the kind of crazy ideas I was suggesting before. These are the three views to put together, but this is something I can skip. The very general message, as you perfectly got it, is that current oncoimmunology is based on a partial and to some extent misleading view of the immune system. Oncoimmunology must be reshaped based on an extended and more adequate view of what an immune system is, namely a control of cohesiveness. One of the most promising avenues, in my view, is to try to develop reparative and not eliminative immunotherapies in cancer. And if I'm right, you see that the iceberg that I presented before, I mean, the lower part of the iceberg is much bigger than just promoting innate immunity about cancer immunology. It would be about rethinking cancer immunology on the basis of repair mechanisms, among other things. And of course, something which is absolutely obvious for people in this room is that conceptual and theoretical thinking in biomedicine is as important as ever. And with this, I want to thank you, and I want to thank the team in Bordeaux. Thank you very much.
Paul. Okay, thank you very much for the interesting talk and the nice hypothesis of the role of the immune system in cancer. And I have so while I agree with you that that continuity and discontinuity and all this dynamic interaction between immune cells and antigens is very important role. So I am with you on this side in a sense. I know that those people who they take this more traditional view of immune system role in cancer, they would insist that it's all about recognition of nonself or altered self. And here they would cite the following evidence for that. Namely, if you block this checkpoint inhibitors, then the immune system will target the new antigens. So in a sense, we have here a case of the immune system that recognizes non-self or altered self, and just out of this polarization state, it's attacking the tumor cells. So how does it fit into this repair sort of framework? I guess that would be my question. And the other is the following, because also I know about your work about microbiota and from philosophical perspective. And here I assume that when we're talking about the role of the immune system in cohesiveness of the organism, you mean the organism also in an extended sense. So we have the extended immune system and the extended organism, right? And the immune system that is also kind of making sure that this whole extended organism is also cohesive, right? So that's also my second question. Thank you.
So for the second question, as you know, the answer is easy. Yes, this is what I'm talking about. The importance of taking into account the heterogeneity of the organism, including the microbiome. For the first question, I think that You're right, but I think what immune checkpoint inhibitors show first is that very often immune systems are tolerant to tumors, which is extremely important for my point. Very often what's going to be created by the interaction between tumors and immune system is a tolerogenic response.
So that I think is very important because for many people, it became very clear through that example that what we should do is to break the tolerance, which I think was a major conceptual realization in the field. Then the question becomes, when we block immune checkpoints, are we necessarily inducing a response to neoantigens? And this is where the objection becomes strong. It's only if we can show that there's a correlation between, for example, the degree and the number of neoantigens and the response that the question becomes an objection. Because otherwise, it's just like, what does it do to block these immune checkpoints? So I think that I've been very interested in that, and I know you've been interested in that as well. It seems to be the case that there is a connection between the degree of expression of neoantigens and the effector immune response that you get. The more that is true, and the more the neoantigens view is likely to be valid. I want to know more about that. I really want to know more about the connection between neoantigens and the effector immune responses. And even if it's true that this connection is strong, it will show us that the more different you are from what you usually are, the more likely you are to trigger an effector response. My take on this is, it doesn't matter whether altered cells should be conceived of as non-self. What matters is the degree of alterations. So if the more altered you are, the higher effector immune response you have, then the discontinuity theory is perfectly able to explain that. So it would be only if there is something about the non-selfness of these neoantigens that I would think that this constitutes an objection to the way I see things. So it's a little bit of a complex answer, but do you see what I mean?
So this is very, very nice and stimulating. I'm thinking, and I'm trying to understand exactly. I fully agree there are components, let's say, of effector cells in the immune system that actually do eliminate whatever that may be, microorganism or cancer, and there are cells which are important in repair, and repair is a crucial component. Now, if we go, I think in infections is different, but let's go only in cancer because that is the argument now. I think that we have to take into consideration when we think, well, can we repair cancer? In the end, cancer results from a genetic component. DNA repair mechanisms are not working properly. There is an accumulation of mutation, and at that moment, actually, control of cell growth is going wrong, basically. Very simple said. I simply do not believe we can repair that. That is so different and so alien compared with a wound. And of course, there are some, let's say you have a basal cell carcinoma on the epithelium, and that looks like a wound and so on. But very aggressive cancers, they are really aggressive. They are cell growth going bonkers, basically, because of fundamental changes in the DNAs of those cells. I do not believe that we can repair that. We simply have to eliminate that. And that elimination is done by the CD8 cells, if it's a proper immune system early enough. And of course, thereafter, it comes a component of repair, which comes in the form of macrophages locally, which start to repair the tissue and so on. But in terms of cancer, I see it very difficult. How can we coexist and repair that? To be very honest, purely because of the alien nature now. And we should not forget, actually, we should not talk only about antigen. We should not forget that antigens come from genetic code, basically. And the genetic code of a cancer cell is so altered, basically, that it can very difficultly, let's say, seen as self, and that we can repair it. And DNA repair mechanisms are not a component of the immune system in general.
Well, that is debated, but I think you're right. We should say that's not part of what we're talking about here, because otherwise it would be too messy. So yeah, thank you. This is a great question with so many interesting components. If you're right, then let's forget about repairing cancer.
I do not know any, no, just very simple. I do not know any simple, not one cancer patient who is cured from cancer, in which cancer cells are still there.
Well, that I think would be...
You can get in a chronic disease model. I mean, I'm a clinician. We have patients that can stay for 20 years with cancer, with a continuous treatment as a chronic disease. But we do not have someone which is just cured and the cancer cell is still there. The cancer cell, in order to be cured, needs to be eliminated.
Well, so for example, many of the New England...
If somebody knows a concrete example, please correct me.
I totally agree with you.
So there's so many layers in your point. So I think first, it's clearly the case that with the immune checkpoints inhibitors papers, some people in the founding papers in the New England Journal of Medicine have big tumors, have very small tumors. The question becomes, what does it mean to maintain a sort of chronic state when we cure someone that has cancer? And I think
it's a very different thing to have a chronic disease and to be cured of a disease.
Yeah.
They're two very different things.
Yeah. So
you can maintain an equilibrium with a disease, including a cancer, by continuously doing things, providing a counterbalance to the cancer process. But you not can cure the cancer by repairing and letting it in.
If curing means eliminating, then curing involves eliminating. If curing means limiting, which is increasingly what people try to do, the detrimental effects of a cancer, then I think we can cure people that would keep a tumor. But that's only one aspect of one. I think this is very important. I think the idea that...
I think it's important, but I don't see as a clinician, I do not see the possibility, conceptually the possibility that we can talk about a cured cancer when we still have to do things and the cancer is still present.
Should we care about talking about the cured cancer, or should we care about sending people back home in a state in a health state that we believe is the best we can reach in a given context?
Okay,
I think this is
very different.
The cancer clinicians I interact with came from a view in which what you should do is to eliminate the tumor, in a view which is much more subtle. I would not say that they would.
Sometimes you cannot do it. There's something else.
And sometimes, again, including with immunotherapy, sometimes people realize that it should not be our aim, because you can sometimes have strong surprises in terms of, for example, disconnection between the size of the tumor and the risk of death, which is something which is very important. So very often now in papers, people will make a difference between what are you checking? Are you checking the size of the tumor, or are you checking in terms of mortality, what's going on with your patients? I think that we should be open to that, and I perfectly agree with you that clinicians at the moment don't want to do that, most of them, and this is perfectly fine. If I were them, I would definitely try to eliminate the tumor and to have my patients happy. But I think this is something we should consider. Should we try to eliminate the tumor to cure people, or to help people who have cancer, if you don't want to talk about curing? Because of course, if it's tautological that curing means eliminating, then we should eliminate. But I think there's really one important question about that. And then there's really one thing to say about repair mechanisms are there in cancer. So then the question is, are they just part of a dysfunctional response to cancer, or is there something else going on? And my sort of crazy idea of the day was there is something going on there that could be manipulated. Again, I have no idea if it's possible, but I think we cannot say the immune response against cancer is purely an eliminative response. This is not what we see. This is not what we see in practice. So understanding what's going on there, I think, is very important. Then manipulating I agree with you, is a completely different matter. But I think that gives us suggestions about what's going on in those responses that should not be neglected.
I agree, but I don't want to.
Yeah.
I have some comments, but I don't want to monopolize the discussion.
So we have a bunch of questions on Zoom, but is that a follow-up?
Yeah.
Okay, so just follow up, and then Judy, and then I'm gonna slow it down, and then we also take questions from the
So it's rather this debate whether there are cancers that are cured but still cancer cells exist, and I'm importing these examples from the screening debate. So whether we should screen for breast cancer, prostate cancer, right? Because it could be dangerous. Like the better the instruments get, the more things that might be problematic get detected. Biopsies can get things into circulation, and you have then this background rate of so many people dying, and they have certain cancers that were never a problem in their lives. So couldn't that be like one source where we say, well, there are these cases where we have this kind of coexistence, we have maybe this kind of successful repair mechanisms that also help?
So I think we need more data about that because people cite always the same autopsy reports with this, prostate cancer and so. And I think there's a lot to say about that, but I think we need much more precise data about that. But you're perfectly right that this is part of what I'm talking about. I've been very much influenced by Mina Bissell in two senses. Everyone has cancer, everyone has tumors, and some of these tumors should count as cancer from a certain point of view. Second, the experiments of Mintz in 1975, old experiment, where you normalize a tumor by putting it in an embryological context, is something I still have in mind. I'm not saying repair is embryological enough to do something like that. What I'm saying is that the normalization through context, which is one of the main ideas of Mina Bissell, could find one possible realization in immunologically mediated repair. And from this point of view, that would suggest that we constantly have tumors and we constantly deal with these tumors through disease tolerance, through repair mechanisms, and they would be dealt with without being too dangerous for the organism. So I should have said that. This is part of my view. Again, it might be completely wrong, but this is part of my view that cancer is much more normal than we usually think, and they can be normalized in all sorts of ways. That's part of my intuition. Might be wrong again.
All right, Judy.
Yeah, obviously fascinating, many ideas going through my head, although I find myself really agreeing, with Mihai. It seems to me that the elimination part is clear. And part of that is because I'm struggling with how wound repair resolves a tumor. What is mechanistically, how does that work? Are you walling it off with collagen deficit? What is the repair doing that's controlling the cancer? I don't know.
So this is entirely speculative. My very general view is that so the very general idea is that in classic injury, you have acute inflammation, chronic inflammation, resolution, and immunity is going to be very important in all of that. So my impression is that very often with those kind of situations, which is exactly the kind of situations we have with cancer, what I do think is that in terms of there is something which is a circle in cancer, and I think either we can try to not allow this circle to start, or we can try to participate in the resolution phase of repair. How would that? I don't know if it could be possible, but the idea would be that it would be exactly. So you see, for example, what we presented here in terms of, the actors of what is that?
Yeah, I like this. I think you need the wrench and the gun.
Oh yeah. I agree with that. But the question would be, how do we use these different tools in different situations? And my impression is that, so the wrench maybe is a limited example for what I'm talking about. I just wanted to talk about this sort of, both functions. But I mean, don't you think that what happens in the resolution phase when tissue repair occurs is something that does not occur in cancer, and nobody has tried to see if we could promote that and see what the consequences would be.
I think because that resolution and inflammation are counter things, right?
Yeah.
And I think the inflammation part is, I still believe, critical for controlling that cancer. And then the repair, and that's why you get this chronic situation where you've got inflammation and repair going on at the same time, and that's a bad thing. But I think if you just controlled the inflammation, which I think you would have to do to get the resolution phase, the cancer would go crazy.
This is exactly my question. So, for example, in terms of what the adaptive immune system would do in that case, I think you're perfectly right. And if you're right, then the idea is dead. If really, I mean, if, for example,
so I think a fundamental issue, something you said much earlier on, is implied that you see the innate and the adaptive immune systems as separate. And I cannot separate the two. They are totally entwined. So I think that what I'm hearing is maybe you're seeing the repair sort of as kind of innate processes.
Not necessarily, because in the actors I showed before, for example, regulatory T cells are important. Gamma delta T cells, which are a little bit in between, are very important. I totally agree with you that the two are intertwined. My question is not, should we stimulate the innate at the expense of the adaptive? It's not that at all. It is, has anyone tried to say we're going to reduce inflammation, we're going to try to promote the resolution phase and see what happens? And again, I'm suggesting to try, those things, certainly not in humans, but just to see what happens in those cases. If you're right that then inflammation, for example, will go down, and then the adaptive immune system will be, the effector immune system, mainly adaptive, will be incapable of dealing with the tumor, then I'm completely wrong, because you would be in a terrible situation. Right. The adaptive immune response in terms of effector response would be very low, and then you would do some fibrotic things on the top of it, so it would be a disaster. I'm just thinking that might be one way in which we reduce inflammation, we promote that resolution phase, and maybe it could be beneficial.
But inflammation is being tried actually to be inhibited, because the inflammatory signal give actually survival signals for the tumor cells. So that there is one actual thing that is trying. For example, Novartis has just done a couple of $100 million study with anti-IL-1 in lung cancer, but I think that if I heard it rightly, they failed basically. So this is actively tried actually, what you are explaining. But what we have to understand, I think, is that in terms of the resolution phase and everything, the mechanisms can be there and the processes can be there, but the same process can be adaptive in one situation and maladaptive in another one. So the fact that it's adaptive in infection, for example, I think your model is working much better actually in an infection. And in certain types of infections for sure, it doesn't mean that it's automatically adaptive also in cancer. It could be, so wound healing can be adaptive for healing a wound and completely maladaptive as a process in a cancer. So this is something.
I agree with that, but my question is, should we divide up wound healing in different subcompartments and different processes? This is my question. So I have no doubt that something can be adaptive and maladaptive in two different contexts. I have no doubt that repair processes in cancer are very often detrimental. I have no doubt with that. My question is is there an avenue within wound healing that has not been tried, where we are thinking about trying to reach a resolution?
I think it should be done together. I don't see it as using it separately, as also Julie said. I think in the end also the process of elimination and repair should be together. So you eliminate something and then you repair. And also in the process of elimination, you need also the process of repair, because if the process of elimination goes on with a lot of collateral damage, then you are in trouble.
I agree with that. My
so
question
that's why you have
I agree with that. My question is, which of these aspects should we try to manipulate? That's the question. So for example, if we say there will be elimination and repair, of course there will. Should we try to promote elimination as much as we can, and then we will have some repair behind that? I think this is the major view at the moment. Let's try to eliminate the tumor, and then there will be repair, of course. My question is that the only way to think about those things? So of course they are connected. Of course it can be maladaptive. I'm just asking a question about, did we try to think about all the aspects of this repair response? And if yes, and if it doesn't work, it's perfectly fine. But I'm still not convinced that this is tried.
Yeah, let's continue this debate maybe over lunch. So there are a bunch of questions on Zoom, and I think it's best to give them the space because they cannot be here with us. So Miguel has a question, and then I'm going to read out some of the rest other questions, and maybe we still have time, but if not, then during the break. So please, Miguel, yeah, go ahead.
Well, thank you very much for a really nice talk. I was wondering whether one should not contemplate in this framework an event that occurs before damage of the cells, which is this stress and damage responses that cells usually undergo in a very evolutionary conserved way once they're exposed, for example, to bystander products of the immune system. So this is before a cell really undergoes full damage and becomes and dies from programmed cell death. And in my view, these stress and damage responses become maladaptive in tumors while they are adaptive in parenchyma cells during infection. So perhaps I'll stop here and I'll let you perhaps reply and see how you put this together.
So I'm sorry, I'm not sure I get the question. So
sorry, sorry. Being at home asking these questions is a bit difficult. Sorry, I'm going to try to rephrase it. So, and in fact, in the context of infection, there's something that I will refer to in the afternoon called tissue damage control, and that's based on stress and damage responses, which are very evolutionary conserved. So this prevents cells from undergoing injury and eventually dying, right? But this is before damage to tissues. This is what prevents actually the real damage. And in my view, these type of processes are actually maladaptive in tumors because tumors use them to prevent the stress and damage that is imposed by the immune system. So it's more of a comment than a question, but we're supposed to discuss.
Yeah, I think you're perfectly right. I think that happens in cancer, and that's going to be detrimental in cancer. I'm not sure it's always going to be manipulation by the tumor, but I think you're perfectly right that in that case it's going to be maladaptive. Yeah, I mean, I would love to talk more with you about this to see what the consequences are in your view about what I presented about immune response to cancer. But I definitely agree with you that it's going to be this sort of damage at the level of the cell before damage at the level of tissue. I think this is indeed a major aspect. Yes.
Okay, thank you.
Yeah, so unfortunately, this noise that we are hearing is going to be here until 2. And there's nothing I can do right now about that. So we have to just deal with that, I guess. Sorry. So the comments. So thanks for the thought, very inspiring. My lecture is the immune system evolved mostly by pressure of the battle, which I believe is an appealing theory. That how does this fit with the existence of immune privileged sites? These theories suggest that the immune system can be very detrimental to tissue integrity and is kept away rather than interacts with our most important organs, tissues. Does this not lead for the immune cell as mostly stability?
So I think the immune system is very destructive. I think the detrimental effects of the destruction initiated by the immune systems are in fact a major issue, major question. Immunopathology is exactly about that. So immune systems are constantly controlled over their effector responses by all sorts of regulatory responses, which happens at all sorts of levels and involves the innate and adaptive immune system. So clearly, I think this is a much more general question than just about immunoprivileged organs. I'm very careful with immunoprivileged organs because I've seen that notion moving from there's no immune response to a much more subtle response where each response is organ dependent and sometimes tissue dependent and is always a process that results from effector responses and regulatory responses, which I think is a much better view than the old immunoprivileged view. But I think the question is much more important than that. Yes, immune systems have evolved not to be too destructive. I think this is super important. So this is, I mean, if that person mentions lymphocytes, it's very different from if that person mentions the whole immune system. So the whole immune system, I think, is the result of many selective pressures, including the need not to self-destroy, if I can use the cell for a second. So I think that is extremely important. And this is everywhere in the organism, not just in immunoprivileged organs. The only difference being that in so-called immunoprivileged organs, we will be more sensitive to the regulatory dimension of that. This is just that, I think. So yeah, I definitely agree with that point.
Great. Then there are two more questions I will read them, and then we need to stop because we are on the time anyway. Thank you for a nice talk with me. Yes, a quick simple question to confirm my understanding. Would you agree that on your view of the immune system, we don't have too strong immune system in case of autoimmune diseases, inadequately detecting and responding to the system?
Yeah, so I mean, I had many interactions with some of you and Gregor Gresslener, who is in Vienna, about what it means to have a strong immune system. I mean, I'm very skeptical on strong immunity or strong immune system. This is a very weird notion that becomes increasingly, I think, problematic. So yeah, I mean, autoimmunity first is normal. Autoimmune diseases are not normal. Autoimmune diseases are not about too strong an immune response. They are about some sort of dysregulation of the balance between different aspects of our immune response. So no, I would definitely agree that we should not define autoimmunity as too strong an immune response.
And finally, thanks for the excellent comment, and please for mentioning the cell culture and the danger theories. Do you think that we approach the science and scientists persist in delaying and building one system theory of the immune system, even knowing that it is about complex dynamic system within living systems?
Yeah, so yeah, I understand this point. I mean, this is also something I've discussed with Bart and others. I think there's really something to say about trying to develop true theories of immune responses. What I mean by that is that, of course, it's a question of system. Of course, it's a question of regulation. Of course, it's a question of dynamics. But just saying that, I think, is very partial. What self, non-self did, what danger did, I think, should be an example for us. They try to explain immune responses and predict immune responses. And this is still something I think is extremely important. So maybe I don't understand the question well, but I think from this point of view, I remain a fan of the self, non-self, and danger in the sense that I think we need to think theoretically about immune responses beyond saying it's a very complex systems, which is context dependent, which is very often what people end up saying when they talk about the immune system. So I would be, prone to insist on the need to still have strong theoretical views about the immune system. And the risk is to be wrong, but this is perfectly fine to be wrong.
Okay, thank you again.